multiple subtypes of pericytes. Initially, in the capillary network, you have what's called an ensheathing pericyte. They have these robust projections that wrap around the vessel, but they have a cell body that sits on top. So unlike vascular smooth muscle cell, where the cell body wraps around the vessel, within the ensheathing pericytes is a single cell body with multiple projections that wrap around. Deeper in the capillary network, you get what's traditionally viewed as a pericyte, in which you have a cell body and these long extensions that go far within the capillary, and they sort of just cover the capillary. And in between the two subtypes, you have something called the mesh pericyte, which is like a transition between the two. And so during my time in the lab, I was focused on this subtype of pericyte called ensheathing pericytes, in particular, the pericytes that were localized at capillary junctions. And the idea here is that these cells, unlike smooth muscle cells, where calcium goes up and the whole cell contracts, pericytes are capable of having independent calcium control, contractile control. So you can have a pericyte wrapping its projections around two capillary branches, constrict one, and allowing blood flow to go in one direction. And to really highlight the importance of this, we did some 2-photon imaging using through our cranial window. So we can inject a mouse with some kind of fluorophore. Here's fixie-dextran to illuminate the vasculature. And this is the movie as well, but what it allows us to do is watch as red blood cells pass through, and we can count the number of red blood cells. And so, using this technique, we can go down to a single pericyte here at a capillary bifurcation and puff on an agonist and cause that pericyte to constrict, all right? And when we do that, we look at the red blood cells passing through each branch. And so when we do that, when we pico-spritz U46619 and just thromboxane receptor agonist and cause that pericyte to constrict, what we saw was multiple things. One is a loss of control. You have blood flow going in every which way. But what we also saw was if you do it right, the pericyte can constrict and actually limit the passage of red blood cells. All right? And this is key because unlike an arterial, artery-arterial, you can put the massive amount of stimulus on and the whole vessel can constrict, well, you'll still have the passage of blood. With a pericyte, you constrict the pericyte and it can completely constrict down to make the passage of red blood cells difficult. And so these pericytes function as this gatekeeper into the capillary network. If you want to get into the capillary network, you want to have nutrient and oxygen absorption, you have to get past the gatekeepers, which are these ensheathing pericytes. And so we wanted to understand better the autoregulation. How do these cells maintain tone to allow for constant blood flow? And so when we talk about autoregulation, there's two arms to it. One is intraluminal pressure, which leads to an increase in mural cell calcium and a contraction. So you increase intraluminal pressure, it's sensed by the mural cell, whether a smooth muscle cell or a pericyte. It increases calcium and the cell responds by constricting, leading to vasoconstriction. On the other hand, you can have flow. Flow or shear stress can stimulate endothelial cell calcium, the production of nitric oxide, and this nitric oxide can now inhibit the production of the increasing calcium in mural cells. And as a result of this, you get vasodilation. And so it's the balance of these two arms, right? You have mural cell intraluminal pressure sensing and then endothelial shear stress sensing. And what's key here, the key signaling molecule here is in endothelial cells, nitric oxide production. And so right now there's a couple of papers that have come out sort of looking at one hand of this process, looking at how intraluminal pressure can increase constriction within pericytes. But on the other hand, very little studies have actually looked at how shear stress of flow can affect endothelial cell calcium. And so when we want to look at flow-dependent vasodilation, we've seen this already today where blood flow can increase calcium inside endothelial cells. The calcium activates eNOS, nitric oxide synthase, to generate nitric oxide, which can lead to the relaxation of the underlying smooth muscle cell, or in this case, pericytes. Recent work has shown that one of the pathways of increasing intracellular calcium is through this piezo-1 channel, where activation of this mechanosensitive ion channel can increase calcium and lead to dilation. And so we wanted to look at the activation of endothelial cells, but in a more physiological context. And so we turned to viscosity. So blood, as we all know, is made up of plasma and red blood cells. Plasma has a viscosity of 1.2 centipoise. Whole blood has a viscosity of about 5.5. So the more red blood cells you have, the higher viscosity it is. And there's a lot of factors that can actually affect viscosity of the blood. In this case, hematocrit for one. The more red blood cells you have, the thicker your blood is. Diet, hydration, medication, inflammation, aging, or in the high-fat diet or the case we're going to talk about. And high viscosity, plasma viscosity, has been associated with a lot of their neurodegenerative diseases or cognitive impairments. Patients with Alzheimer's disease or vascular dementia have showcased a higher blood viscosity. With aging, there is a correlation between aging and higher viscosity of blood. And so we wanted to look at how viscosity affects this ability to the vasculature to respond to increases in pressure and increases in flow. And so the preparation, or the model that we use, is actually the pressurized retina preparation. In this preparation, we can cannulate the feeding ophthalmic artery, pressurize the whole tissue, and look at pressure steps both in the arterioles, radiating arterioles, and the capillaries within sheathing and instrument pericytes. And this was done by my graduate student, Christian, who just actually defended it. And so the first set of experiments we did is using a cadherent GCAM6F. So this is a mouse that expresses the calcium indicator just in endothelial cells. And we can look at the response to calcium as we increase flow at different viscosities. And to summarize this in short, what we saw is that if you use PSS, a 1 centipoise solution, you get these transient calcium events. If you increase the viscosity to a physiological 1.6 or almost pathophysiological 3 centipoise, you get a transformation of the calcium signal from a lot of little calcium signals to one robust signal, right? One robust calcium signal. And we understand that calcium in endothelial cells leads to nitric oxide production. And so we wanted to see what's the effect in the circulating mural cells. And so for this, we used another mouse. This is an NG2 GCAM6F mouse. So this is producing a calcium indicator in mural cells, arterioles, pericytes, and we do a simple, increase in pressure or flow at different viscosities. And so if you use a PSS, you get this very robust calcium response in the mural cell. And as you increase viscosity, you would predict you get a decreased response in the mural cell. And this is what we see. So as you increase viscosity or flow to 1.6 or 3 centipoise, you're decreasing the mural cells constriction. They're dilating. And if you use L-name to block nitric oxide synthase, you return the massive calcium response. And so in the capillaries, much like arterioles, there is a communication mechanosensation between endothelial cells and mural cell activity. So again, mural cell intraluminal pressure leads to mural cell calcium. Increased plasma viscosity can lead to increased shear stress in endothelial cell calcium and nitric oxide production, and the inhibition of the mural cell calcium. And so we wanted to see how does a high-fat diet affect plasma viscosity and affect this mechanosensation within the vasculature. And so mice were given a high-fat diet that did not lead to obesity. That was important for us. We didn't want to complicate it with obesity. We just wanted to increase the intake of cholesterol to increase the viscosity of the blood. And we did that. With these mice, if you look at the viscosity of the blood, it went from 1.2, 1.3 up to 1.5 centipoise. And as a result, we wanted to look at the myogenic response at different intraluminal pressures and viscosities between the arterial, the ensheathing, the transition segment, which has the ensheathing pericytes, and the capillaries. But let me take, this is a lot, so let me take you through it slowly. I don't know if you can see there, but in the arterial, when mice were given a high-fat diet, what we saw was an increase in myogenic tone, an increase in the contractility of the vascular smooth muscle cell. And this sort of makes sense. If you're in a chronic condition of high viscosity, high endothelial mediated generation of nitric oxide, the smooth muscle cells respond by increasing their contractility. And so you get this robust increased constriction when in PSS, so when during low stimulation. Interestingly, we did not see that kind of remodeling occurring within the transition segment or within the capillaries. And if you looked at the arterioles and you increase viscosity, what we saw was that in the high-fat diet, actually in both controlled and high-fat diet, you were able to decrease the tone generation. Again, suggesting an intact balance between the endothelial-mediated nitric oxide production and the pressure-induced vasoconstriction. So what about the pericytes? Remember, there was a lack of remodeling in pericytes in the high-fat diet, but as you increase viscosity, we saw a different trend, right? Unlike the control conditions or in the arterial, where as you increase viscosity, you decreased the generation of myogenic tone. What we saw instead was viscosity had little or opposite effect in the high-fat diet within the capillaries, suggesting a different form of vascular remodeling occurring within the capillaries versus the arterioles, whereby the remodeling not increased the contractile tone of the mural cells, but rather decreased the sensing of viscosity or sensing of shear stress in endothelial cells. So what we showed here is that viscosity shifts the transient signal from a transient calcium signal to a sustained signal. This signal led to an increase in mural cell dependent tone and calcium, and that high fat leads to vascular remodeling. In the arterioles, it leads to an increase in contractile tone, but in the capillaries, it led to a decrease in sensing of shear stress. So how does this difference affect how blood flows into the capillary network? And to do this, and I don't think this movie will show it. So in this example, we just put a dye in, and you can see the dye flow through the capillaries. But one of the key things here, if you take a look at where the little yellow mark is, you see flow go into the capillaries, and then a delay, and then opening into our capillary network. So there is some kind of regulation. It isn't just passive flow like a river flowing down a river, water flowing down a river, but there's an actual gating mechanism occurring within the capillaries. But these experiments are difficult because you only get a one-off kind of experiment. So rather, we turn to fluorescent microspheres. And so here in our cannulation, we can introduce fluorescent microspheres and watch how the fluorescence goes through the capillary networks. As you can see there, you can see the fluorescent spheres go through all the capillaries. And so we wanted to look at how blood flow entered the capillary network with different viscosities, and under control conditions with 1 centipoise or PSS, you get random distribution. Flow is going in the capillaries, high flow in the capillaries and low flow in the capillaries. But once you get into a more physiological level of viscosity, you had a more regulated flow control. And then if you increase viscosity to a little pathophysiological, you get limited flow to the capillaries. And then in the high-fat diet, we lost all control. So this suggests with the high-fat diet, the loss of this endothelial capillary-mediated sensing of shear stress led to a decrease in actual perfusion of the capillary network. So in conclusion, the high-fat diet increased intraluminal viscosity and disrupted microvascular signaling and remodeling. And this led to a diminish in the flow to the capillary network. And so Zungil asked to talk about potential projects when it comes to nitric oxide. Because health cognition depends on the precise regulation of cerebral blood flow, meeting that specific metabolic demand, this viscosity-driven disruption of the microvascular can accelerate progression into cognitive decline. The inability to get red blood cells, the oxygen and nutrient delivery into the capillaries and into the neurons can limit the neuron's activity. So the overarching hypothesis would be that high-fat diet drives cognitive decline by inducing plasma viscosity-dependent disruption of the endothelial to pericyte mechanotransduction, leading to chronic cerebral capillary hypoperfusion, neurovascular uncoupling, and neural dysfunction. I won't go really into specifics, but basically what we want to look at is sort of the mechanism, how high-fat diet decreases the endothelial or alters endothelial nitric oxide signaling and the pericyte-mediated mechanotransduction. And then we can target piezo-1 or the components of eNOS coupling or the mechanics of pericyte dilation. And then the next, we would like to look at nitric oxide dependent microvascular signaling, how it regulates cerebral capillary perfusion, flow patterns, neurovascular coupling under the high-fat diet. So this is taking more in the physiological, translational aspect of it. How does all this affect how capillaries perfused? And because we have the tools of this ex vivo retinal preparation and in vivo 2-photon imaging, we can really look at flow dynamics. And the idea here is that with normalized or supplementation of nitric oxide signaling, we can open up these gatekeepers, allowing for proper capillary perfusion. And then lastly, you can't study cognitive decline without looking at sort of the causal link of this. And so the idea here is that if you don't get, or you have chronic capillary hypoperfusion, you can drive cognitive decline. And then for these experiments, it would depend on behavioral testing, looking at episodic memory, working memory, and spatial learning memory in mice exposed to a high-fat diet. Now, that experiment has been done multiple times in the literature, but in this context, we want to look at whether nitric oxide targeting intervention can increase or address these cognitive declines. All right, so that's it. I'd like to thank Zungil and everybody here for your attention, my lab, the people that actually do the work, and also the funding right now. Thank you.
All right. Gonzalas, so capital.
Wonderful work, Albert. As you know, people have been using the CGRP receptor drugs for migraine. Generally, it's thought to be targeting capillary functions and capillary dysfunctions. What does CGRP effect look like in both your system, as well as if you do this experiment in a eNOS knockout mouse? Does it supersede the effect of eNOS?
Yeah, no, those are good questions. I mean, that's something we can address. Zungil is trying to generate these mice, these knockout nitric oxide knockout
Right.
mice.
That's
and so we would be able to see that effect there. Again, a lot of what our understanding of the nitric oxide signaling in parasites has always been they're like smooth muscle cells, so it has to be the same as smooth
Exactly.
muscle.
Yeah.
No, it doesn't have to be the same. I mean, we're showing that the sensing parasites have a different sensing of mechanotransduction than smooth muscle cells. They are a different cell type.
Interesting. The other thing is, just like in the brain where you have periods of or regions of quiescence and regions of activity, the same happens in the gut.
Yeah.
Where you will have not all neurons light up at the same time. Neurons that remain quiescent for a very long time, neurons that are actively dying, neurons that are being constantly reborn. Size of ganglia are very different, and if you actually look at any of the images that we generate, the amount of vasculation that comes extremely close to mesenteric ganglia is just huge. The other difference between mice and humans is that in mice, the vasculature goes around the ganglia. In humans, the vasculature goes through the ganglia, which is very interesting. Which suggests that there is some kind of a role that they are playing. Do you see similar mechanisms of gating exist in the gut?
We have just started looking at it. We've looked at parasites and this idea of ensheathing parasites in the gut, but we haven't really. They have myogenic tone, pressure-induced tone. They showed a branch-to-branch deviation of flow, but we don't know to what signaling or in response to anything right now.
What I would suggest is that look at this in conjunction with mitochondrial load, because what we see is that there's a huge diversity in abundance of mitochondria in neurons. So not in the colon, but in the small intestine, neurons have the most amount of mitochondria.
Yes.
And B, not all neurons are enriched in mitochondria. So would suggest having a look at that in whether there is more blood flow going towards regions that are low in mitochondria. That would be my expectation, because they will be more glycolytic as compared to those where there is a higher amount of mitochondria, where there will be a lot of lactate shuttle.
Yeah, no, there's been a few studies looking at sort of correlation of capillary density in these regions, different neuronal regions, and they sort of correlate with it. The more mitochondria you have, the more capillaries you have.
Right.
The brain is interesting not only because of the heterogeneous need, but you're in a fixed volume of space. You can't just overperfuse the brain because it's in the skull, and you can increase intraluminal parts. So the vasculature has to do more with less, basically. Unless you're in a region where there's a higher demand, you increase the number of capillaries you have.
And then do you see similar mechanisms for lymphatics?
No, we haven't really looked at lymphatics. That would be interesting, or the glial lymphatic kind of thing, but we haven't looked at it.
Thanks.
Did you check the triglyceride level in the blood after high fat diet in those mice?
That's in the next grant. Yeah, we're going to look at more of the compounds. This was sort of just giving it a high fat diet. We have another story where we're trying to look at cholesterol within endothelial membranes, and that cholesterol functioning as sites for amyloid beta genesis. And in that context, we want to see more of the lipids within the blood that contribute to that.
As a matter of fact, just one more thing. So as a matter of fact, the hypertriglyceridemia induced pancreatitis is very common, especially in China. So I think that's associated with not only genetic mutation, but also high fat diet in those populations. So I think definitely there's a significant collaboration potential can happen between my lab and your lab in the future.
Yeah. I mean, there's a lot of interesting things, and there's a lot of things that increase viscosity that we didn't really think about. Hypoglycemia increases viscosity, age increases viscosity. So we wanted to look in that kind of context. But we want to know what's inside the blood.
Yeah, great talk, Dr. Robert. So my question was that why you choose the high cholesterol diet, and how long you feed the mice, and why you avoid obesity? I'm thinking
We didn't want to complicate so originally those experiments were done for another project, which turned out to have no difference. And so we used those mice.
Should be very similar pattern, right? In obesity and just feeding with high cholesterol diet.
But the obesity has compounding effects that we didn't want to introduce.
That's why I want to know, what confounding things.
Well, obesity in general can lead to a lot of more downstream.
But those patients also have
chronic
the high
disease.
viscosity, right? Even
We just want a simple model of high viscosity.
Yeah. Just want to curious. Thank you.
All right, thank you. Last question. Do the parasite like love? Because they hugging endocytes. You see the like little picture, like like this hugging. They are wrapping around, wrapping around endo.
Capillary, yeah. During my grad school years, I had a funny picture. I put my mentor at the time hugging a vessel, just like a parasite.
Parasite.
Oh.
That was great though. Thank you. That's good. Thank you.